Okay, hello and welcome. Today I'm going to be going over the 2024 AP Biology FRQ solutions. I'm going to go through all 6 of them in a couple different parts of this video. So we're going to start off with number 1, and just keep in mind that they haven't released the solutions or the marking criteria for this just yet. So these are what I personally think are the answers. They may accept a wider range of answers. We just have to wait till the marking scheme comes out. But I'm going to start with this first one. They did slightly change the format of the questions. They are not giving you the full question and then giving you like the part A, B, and C afterwards. So we're going to do this in sections and then go through. I'll give you as many possible options as I think that they'll accept. But again, there could be multiple answers for these. Just keep that in mind. Okay, so we're going to start off with number 1, which is one of the bigger questions. These are usually about 10 marks. Number 1 and number 2 should be about 10 marks. The other one should be probably around 4 to 6 marks each. So to start off with this first one, the start of the question says, crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. So this first sentence is actually very important. It goes along with obviously this question that you can see here, but this kind of sets the stage for it, and this is going to be fairly important for the last part of the question. Now, A number 1 says, describe the function of the S phase in interphase. Now, this is a very straightforward question. There's no tricks in this. This is just general knowledge. So this is talking about your stages of the cell cycle when you're looking at not just meiosis, but the entire cell cycle. So I'll draw a diagram and I'll tell you guys what you should put. So this is the entire cell cycle. You have a sliver of it that is for meiosis, or what's known as the M phase. So this is the actual division phase here. So within the M phase, you have prophase, metaphase, anaphase, telophase. And then after that, you have your G1 phase, which is your first growth phase. And then you have the S phase here. And then you have the G2 phase here. Now, from here all the way around to here, so basically everything, not including mitosis or the M phase, is going to be interphase. So all of this is interphase. It's basically when the cell is not in its actual division process. So we're looking at this part right here, the S phase of interphase, and it's just asking for the general function. So I recommend you write full sentences, but just for time purposes, the function of the S phase is DNA replication. That is the only function of the S phase. Very straightforward. I would just write full sentence saying the function of the S phase in interphase is DNA replication. I always remind my students that S, I usually remember, stands for synthesis, which is the DNA synthesis. So obviously you're going this way. So before you go through meiosis, which is when you obviously split into multiple cells, you do need to first replicate your DNA so that you make sure each one of your cells has the proper amount of DNA, the full 46 chromosomes if you're human. So the S phase is where the division of the DNA happens, and then you get the division of the actual cell in the M phase. So this is the first one. Okay, so this is the next part of the question. I'll read through the whole thing and I'll highlight some important parts. So this one says, some regions of a chromosome called hotspots display a higher frequency of crossing over than other regions do. So this is something that we briefly go over, but they do give you the definition here. So we're looking at places where crossing over happens at high frequencies. So crossing over is suppressed in chromosomal regions near the centromeres. The centromere region of a duplicated chromosome includes a collection of proteins that form a structure called the kinetochore. You learn about all of this in Unit 4. Scientists hypothesize that one or more of these kinetochore proteins are responsible for suppressing crossing over around the centromere. So they believe that around the centromere, which they give you a diagram here, the crossing over should be suppressed. So there should be less crossing over near the centromere, likely just because there are like bulk proteins there. So to investigate their hypothesis, scientists modified chromosome 8 in yeast such that in one cell, one chromosome from the pair of homologous chromosome 8s contained the gene encoding red fluorescent protein, also known as RFP, while the other chromosome formed a pair containing the gene encoding green fluorescent protein, or GFP. the cells expressing RFP emit or also give off red light, and then the cells expressing GFP emit a green light. The models of the modified chromosome 8s before and after crossing over are shown in figure 1. So they do give you a figure in this one. Always read your figure captions. It says, models of modified chromosome 8 used in the experiment A before and B after crossing over occurs at the hot spots. So this is before crossing over. You can see that you have 2 homologous chromosomes here. Again, always remember your difference between homologous chromosomes and sister chromatid. These are homologous chromosomes. One of them has RFP, the red light, and then one of them has GFP, the green light. And then when they do crossing over, you guys know that the 2 inner sister chromatids are going to swap, and they're going to swap somewhere within this hot spot, right? Because we said it's going to be suppressed somewhere near here, but there is going to be a lot of crossing over in the hot spot. So after crossing over, there's likely a couple different combinations of what could happen here. However, like depending on how high up you're doing the crossing over, but they just give you one version here where you had crossing over of those 2 inner sister chromatids that are attached to 2 different homologous chromosomes. And again, homologous chromosomes are not identical. So you did have crossing over that caused this RFP chunk here to cross over and swap with the chunk that is here. And then you end up with this inner sister chromatid having nothing because it gave the RFP away during crossing over. And then this inner one here had the GFP and the RFP because it maintained it or gained it during that crossing over process. Okay, so this next part here is not actually about this diagram. It's a pretty general question. It asks you to explain why some haploid cells formed after meiosis in the experiment will only have one fluorescent marker. Okay, so they'll only have one fluorescent marker, meaning after crossing over when we're looking at this, they will only have either GFP or RFP. Now, it won't be all of them. As you can see, some of them might have 2, some of them might have none, but some of them only contain one of the markers. And the reason for that is if we're looking at meiosis, meiosis has 2 steps. So for meiosis, you have 2 divisions. The first one is going to be your division of homologous chromosomes. So this is after crossing over. So you do crossing over and then you separate your 2 homologous chromosomes. So one cell will contain this chromosome and one cell will contain this chromosome. And then after, you have meiosis 2, which separates your sister chromatid, which actually breaks the arms off like this so that you only get one strand of this chromosome. Now, that is the reason that they may only have one marker, because after meiosis, you're not ending up with a full duplicated chromosome. You're only ending up with half. So if we're going by this model here, you should end up with 4 haploid cells, as you usually do with meiosis. This one should contain, one arm of the chromosome with the RFP. So we'll put a little RFP here. And then one of them should contain this one that has no signal. The next one over here should contain one here that has 2 signals, the GFP and the RFP. So we'll put an RFP marker here and a GFP marker here. And then the final one, which is this last chromosome arm here, will go into its own gamete, and that will actually only have the GFP. Okay, so just due to how the chromosomes separate during meiosis, and it's really not that specific to this question, you just need to know that at the end of the day, when you finish meiosis, you're going to end up with 1 chromosome in each haploid. And since for the haploid cells, you can have one of these markers. So these 2 both have only 1 marker. And so you didn't have to draw all this or write all of this. Frankly, the answer for this one, what I would put, so it just says explain. So you only really need 1 sentence for this. Explain why some haploid cells formed after meiosis in this experiment will have only 1 fluorescent marker. So for this one, the reason for that is that you're going to have haploid only contains haploid 1 copy of each chromosome. And you can also say because there's 2 divisions, You can keep expanding on this. So overall, haploid cells, which are what you get after the end of meiosis, is 2 divisions, and then you're going to end up with only one unduplicated chromosome in each one of your gametes. That's why some of them only have one marker. They're not getting both of the duplicated chromosomes, they're only getting one, so one marker. Not for all of them, but some of them do, like they mentioned here. So it's just because there's 2 divisions and you're going to end up with one chromosome in each of your cells. Okay, so that is A number 2. And then moving on, you have another graph here. And in addition to the question, it says the scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a known crossing over hotspot on chromosome 8 affected the frequency of crossing over at this location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore proteins attached to the hotspot. So right here, they're giving you the parameters of the experiment. So group 1, no kinetochore proteins. Group 2 contained kinetochore protein CTF attached to the hotspot, and group 3 contained kinetochore protein IML attached to the hotspot. You don't need to know anything about these proteins. They're just telling you that those are the conditions. So for each group, the scientists determined the frequency of crossing over between the RFP and GFP genes. To determine the frequency, the scientists added the number of cells emitting both red and green light to the number of cells that emitted no light and divided by the total number of cells. Now, this part probably confused a lot of students here, but frankly, it's not very important for the question. They're just telling you how they got the metric over here, which is the frequency of crossing over. But frankly, you can just take that at face value. You don't need to think about any of like the light emitting or anything like that. So if we look at our diagram here, again, always read your figure captions. It says the frequency of crossing over in a hotspot on yeast chromosome 8 for cell groups treated with different kinetochore proteins. Error bars represent plus or minus 2 standard deviations. Okay. So you have your x-axis, which is attached kinetochore protein. You have 3 groups, which we've already mentioned here. You've got your no kinetochore proteins, CTF kinetochore proteins, and then IML kinetochore proteins. And then on your y-axis here, you have frequency of crossing over as a percentage. Okay, so let's go into this. B part 1 is very straightforward. It says identify the control group for the scientists' first experiments shown in figure 2. So they're asking what is the control group for this. So this is a very straightforward question. You guys should remember that control groups are what you're comparing it to. Now in this experiment, you're trying to see the effect of having different kinetochore proteins attached to that hotspot. So your control group, what you're comparing it to, is going to be the one that has no kinetochore proteins. So your control group is actually going to be the one that has no proteins, because you're going to take your CTF, you're going to compare it to this, you're going to take your IML, and you're going to compare it back to this to see if there's a difference. So your control group, all you would have to say is the group with no kinetochore proteins. Okay, so that one is pretty straightforward again. If we go to part 2 of B, you have a follow-up experiment, which says, in a follow-up experiment, the scientists created a modified version of CTF in which the DNA binding portion has been removed. They compared the frequency of crossing over in yeast cells in the presence and absence of unmodified CTF with that in yeast cells in the presence and absence of modified CTF. The data isn't shown. In the follow-up experiment, justify why the scientists use a modified CTF protein that is unable to bind to DNA as a control. Okay, so they don't give you any data behind this. They're just asking you the reasoning why you would want to use a CTF protein that can bind and can't bind. Okay, and the reason for that one is it's also just going to be a control. So the whole point of these kinetochore proteins is that they physically bind to the DNA to affect crossing over. Okay, so if you have one that is actually physically unable of binding to the DNA, that's going to be a really good control. So they use the modified CTF protein that can't bind, and then they use the regular CTF protein that can bind. So therefore, you can see directly as a control of comparing both of them, whether DNA binding is something that's affecting the crossing over frequency. So for this one, you can do a couple different options that I've seen. Is it allows for a direct comparison because, again, it is going to be a control. So you're allowing a direct comparison because you're exposing both to CTF. However, one is not actually binding. So that's actually going to be a much better control than the no kinetochore proteins in terms of a direct comparison. So it allows you to directly compare and see if DNA binding is affected. DNA binding affects crossing over. There's probably a couple different ways in which you can say this, but overall it's just used as a control. And the main point of controls is going to allow you to make a direct comparison and see, specifically in this example, if DNA binding affects the crossing over, or really how the CTF protein in general, because the one that's modified shouldn't bind in the first place, just seeing a better comparison of what it's actually doing. Okay. And then 3 is asking again the independent variable in the follow-up experiment. So the independent variable of the follow-up experiment. So make sure you're not doing the independent variable of this one over here. This is our follow-up experiment. So for this one, you're taking modified CTF protein without DNA binding and then regular CTF protein, and you're looking at frequency of crossing over. Your independent variable is always your x-axis, so the thing that you're changing. So your independent variable is whether or not the CTF is modified. You can say this in a couple different ways. So if the CTF is modified, you can say, if the CTF has the DNA binding portion or not, anything like that, but that's what would be on your x-axis for this one. Your y-axis, your dependent variable, is your frequency of crossing over. Okay, so that is going to be B. Now, if we move on to C, it says, based on figure 2, describe the effect on the frequency of crossing over when CTF is attached to chromosome 8 compared with the effect when IML is attached to the hotspot. So that takes us back to figure 2. You're basically just looking at the effect of CTF and IML. So we'll take a look at this. And again, like I said, it's really important to establish in this part right here which one is the control, because you're ultimately going to be comparing it back to this to see if it's increasing or decreasing. So for this one, you can see that CTF is decreasing because this is with no protein. And then when you attach CTF, there's a decrease in the frequency of crossing over. You can also mention that it's significant because if you look at the standard error bars, there's no overlapping. And for IML, there is no change. So they're pretty much even when you look at the levels of crossing over for no protein and IML. So there was no effect. So I don't know if this would be 1 mark or 2 marks if you needed to say both of them separately. So for CTF, it would have decreased crossing over. And again, write these in full sentences. And then for IML, there's no change. And then I would maybe mention that it's significant, just in case. Again, I don't have the marking scheme for these, so I put as much information as you can. However, you likely wouldn't get a mark off if you didn't mention that it was significant. I'm just putting that just in case. Okay, so now for part D, it says predict the effect of the number of copies of chromosome 8 likely to be present in the resulting daughter cells when CTF is attached to the hotspot. Okay, now this one is the rather difficult one, and it requires you to go all the way back to what I highlighted at the very beginning. So for D part 1, so we're looking at the number of copies of chromosome 8 likely to be present, resulting in daughter cells when CTF is attached. So we've already mentioned that CTF should decrease crossing over, okay? And part 2 is also providing reasoning, so I'm just going to answer these all as one. So I'm going to tell you and then provide my reasoning. So CTF decreases the number of crossing over events. What would that have to do with the number of copies that are distributed during meiosis? So this takes us back to here, which says crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. So we've established that CTF decreases crossing over. Now, if crossing over is required for proper division, decreasing crossing over should decrease proper division, and it should decrease the accuracy of the divisions. Okay, so for this one, I would say, so predict the effect. In terms of the number of chromosomes, overall, I'm pretty sure there could be a couple different answers for these, because overall you would just get non-disjunction. So you could say either that some cells would have more or less. So +1, -1, overall they would be a non-disjunction event. So you can't say for certain. They probably would have accepted both, or you might have need to say both, but non-disjunction would occur. So you'd say non-disjunction, where some have extra and some are missing. And the reason for that, so my justification is that CTF decreases crossing over. And then since it's required, for proper distribution of chromosomes, you're going to get non-disjunction, or basically less accurate moving around of those chromosomes. So this is the bulk of the question, that you have some with extra and some with missing chromosomes. And then again, the reason for that is since CTF decreases crossing over, it should also decrease the accuracy, or how well the chromosomes are able to separate, and that's going to give you your non-disjunction. Okay, now for Part 3, it says, explain how the presence of hotspots, seen in Figure 1, could increase the likelihood that a population will survive in the presence of selective pressures. So this one actually doesn't have to do too much with the question. We need to know the purpose of crossing over. Now, we know the purpose of crossing over is going to be for genetic variation, and that's actually also the reason that certain populations survive in the presence of selective pressures. So we know, and I think we'll come to a question that also addresses this, that low biodiversity or low genetic diversity is not great for populations or ecosystems, because if there is some sort of pressure, or there is some kind of disturbance, they're going to be very hard to survive and bounce back from. So what you want is genetic variation, and that's what crossing over's purpose is. So the presence of hotspots would be good, because I'll just pop this up here. So increased crossing over, or hotspots, increases genetic variation, which should result in higher resilience. Again, you didn't need to put this exact wording, but the general gist of the fact that crossing over is going to increase genetic variation, and we know genetic variation is going to help populations survive under some sort of pressure. Okay, so this was number 1. It wasn't too bad. Here are all your answers for them. This one was probably worth about 10 marks. Okay, so this is number 1. Okay, so now to get into number 2, this one says, to investigate how increases in environmental temperatures affect the metabolism of certain organisms, researchers incubated liver cells from toads at different temperatures and measured 2 markers of metabolic activity seen in Table 1. You have rate of oxygen consumption and rate of ATP synthesis. Okay, so here's your table here. You've got your 2 metabolic markers, rate of oxygen consumption, rate of ATP synthesis. And then you have them at different temperatures. You've got 20, 25, and 30 degrees Celsius. Okay, so part A actually has nothing to do with this table. It's a pretty general question, just like the last one, which says, describe the role of water in the hydrolysis of ATP. Now, you guys should know the role of water in like a hydrolysis reaction in general, but they wanted it to be specific to ATP. Now, the big thing that happens with ATP is that ATP is converted to ADP, and that's how the energy is released. So you have an adenosine here, and then you have 3 phosphates attached to it. 2, 3. And then when you add water to the equation, like this, what's going to happen is you're going to cleave this bond here, just like it does for any other macromolecule like carbohydrates, proteins. The water is going to split that part, and then you're going to end up with ADP, so adenosine diphosphate, with a loose phosphate here. Okay, so if I were to actually write that out, the role of water is to break, I'm not entirely sure how specific they want you to go with this, so I'm going to say break the covalent bond, because it is a covalent bond, between the third and second phosphate. This is going to release the energy, and what it is also going to do is it's going to attach an OH to one of the molecules and a hydrogen to the other, and that's how it facilitates. So OH on one of them, and then H on other. That's how it breaks that bond, by giving one an OH and one an H. They have their full octet, so they're not going to have to make that bond anymore. So again, don't know how specific they would want it, but this is the answer to this question. So breaking the covalent bond between that third and second phosphate, and in addition with H2O, you're going to have that break apart into OH and H and attach to one here and then one here, and that's going to break a covalent bond. Okay, so the next part says, for part B, it says, using the template in the space provided for your response, construct a bar graph that represents the data shown in table 1. Your graph should be appropriately plotted and labeled. Okay, so they tell you what type of graph to use, which is very beneficial. And so you're going to be doing a bar graph with table 1, which is right here. They had you use the template that was given. I don't have the exact template, but I was told that it's about 20 by 30. So we're going to zoom this in, and we're going to construct this bar graph. Now, what's really important for these are identifying what your X and Y axes are, because most of your marks are going to be from actually just labeling the graph and constructing it. So your x-axis is always your independent variable, so that's the thing that you're changing, okay? And then your y-axis is the result, or what you're looking at. So in this case, our x-axis is going to be temperature, okay? Because that's the thing what we're changing. We're trying to see how the change in temperature affects our metabolic markers. So you're going to have temperature down here, and then you're going to have your metabolic markers here. So there's a couple different ways you can do these, because you do need to plot oxygen consumption and ATP synthesis separately. I was looking, and I saw some people had opted to do like a double bar graph. There are a couple different ways to do this. I'm going to show you guys the most simple option and what I feel like most students would have done. But again, there are probably a couple different ways if you wanted to do like oxygen on this side and then ATP on this side. It does just get an extra complicated. So I'll show you the easiest way that I would recommend doing it. So for this one, I would put on your y-axis your metabolic marker. So again, always important to put your titles here as well as units. So metabolic marker, and then please, please, please put the units. You won't get a mark unless you put the units. So it is in nanomole. Again, you can just copy it directly from the table. Milligrams of, and please write the whole thing, mitochondrial protein, and then plus minus 2 standard errors. Okay. Alrighty, so they usually don't give you marks for putting a title on the top, but if you were to put a title on the top, you could just copy this right here, rate of oxygen consumption and ATP synthesis at different temperatures. Maybe put it just in case, but they usually don't give you marks for that. Now, in terms of how I'm going to set up this graph, you have your 3 different measurements. So you have 20 degrees, 25 degrees, and 30 degrees. Okay, so let's do this. We'll put 25 degrees here. It's just most logical. Oops. Okay, actually, backtrack that. So the best way to do these is I'll put 20 degrees Celsius here, I'll put 25 degrees Celsius here, and then 30 here. And then for these ones going up, it's really important that you choose the correct scale. So again, I believe that they gave you a 20 by 30. So there's a couple different scales in which you can do this. As long as it makes logical sense, they'll give you the mark. So I would say if they gave you 30 on the y-axis, you could either go up by 1 or by 2. Either would have been fine. And what you could have also done, which they don't really teach you, and would have also been acceptable, is because the first number doesn't start, at 1, the first lowest number starts at 12.6. You could also start your graph at, 12. Okay, and then just go up by ones for here. You usually put a little symbol here that just indicates that you skipped a chunk. But just for the sake of this, I'm going to just do the whole entire graph. But again, if you did that little skip, as long as you plotted it correctly, you would have been fine. So I'm going to go with the fact that each one of these squares is 1. So you don't need to plot everything. So 2, 4, 6, 8, 10. And then our highest number here is 22.1, so I'd probably just go up to 24. You can do it all the way to the top. It's not completely necessary. Okay, so now let's start with our first numbers. So we're going to start with our 20-degree measurements. Okay, so these are our 20 degrees measurements from the tables. You've got the first one at 12.8 and the next one at 12.6. Okay, so for these ones, you're doing a double bar graph. So you're stacking them beside each other. You could also technically stack them on top of each other, or again, like I said, do a double bar graph with oxygen on one side and ATP on the other. This is just the most simple way to do it. However, if you do it this way, you need to include a legend, and I'll show you. So 12.8 for oxygen consumption. So this won't be the most accurate graph, but if you have 12 here, 12.8 should be around here. You can make them single bar, double bar, it doesn't really matter, whatever works best for you. So I just did 2 across just to make them a little bit easier to see. And then the other important thing is you have to put your standard error in. So here it's 2.2. Now they won't go right up until the millimeter, so as long as you're pretty close with it, so 2.2 is going to get you to 15 upwards, so you're around here, and then 2.2 down gives you 10.6. Yes. Okay. And then your other one is 12.6, so we'll put it slightly lower. Again, as long as it's somewhat accurately plotted, they will give you the marks for it, and then plus or minus 1.6. So we'll go up 1.6 here, and then 1.6 down. Okay, now just to make sure that you can distinguish between these two graphs, because they are 2 different measurements, one's for oxygen and one's for ATP, you want to either color one in or put a pattern on one of them. I'm just going to color in one of them like this. This would have been more than enough, and leave the other one blank just so you can distinguish between them. And then we'll put the legend in as well. So for 25 degrees, we've got 16.5 and then 16.8. So these are fairly close numbers. I don't know if they allow you different colors, but simply coloring in one or like putting hatch marks in it would have been fine. So you've got 16.5. For this one over here should be around, oops, make it a little lower. 16.5 should be around here. And plus or minus 2, so it should be here, to 18.5, this, and then 16.8, we'll put it slightly higher, and then plus or minus 2 again. And then making sure to color in the one that is for oxygen consumption. And then finally, your 30 degrees, you should have 22.1 and then 21.07. Okay, so 22.1 around here. And then plus or minus 0.7, so it's a little bit smaller. Again, they're not going to measure it to the millimeter, but as long as it falls within an appropriate range, it should be fine. So that's about 0.7. And then 21.07 should be pretty much like the exact same. Or sorry, oh no, this is not 22.07, it's 21.07, my bad. So 21.07 should be one lower. So right about here. And then plus or minus 0.8, that. Okay, and then drag that down, and then making sure to color in. So these ones have smaller error bars. If you were to draw bigger error bars for these ones, they would mark you wrong. So just keep them relatively proportional. So then you have to write a legend if you're going to do it this way. Frankly, whichever, like unless you do the oxygen on one side and ATP on the other, you have to put a legend, especially if you're also doing a stacked bar graph. But this is the simplest bar graph that you can do for these ones. And I always say make it as simple as possible for these ones. So you would do a legend where you put this little colored in one, and then you would put, we made this one rate of oxygen consumption. So rate of oxygen consumption. You don't have to put the units here because you already put them here. You can just in case, but all they really want to know is which one's oxygen and which one is ATP. So this one's going to be rate of ATP synthesis. Okay, so that is this one. So you will get marks for properly labeling the X and the Y axis. So you should have temperature, and then you should have metabolic markers with the units. You need to accurately plot these with accurate error bars. And then if you do this type of graph, you have to make sure that you include your legend as well. So this is it all graphed. Okay, so that takes care of number one. Number two says, based on the data provided, determine the temperature in Celsius at which the rate of oxygen consumption is different from the rate of oxygen consumption at 25 degrees Celsius. Okay, so they're just asking you to determine, so they don't need an explanation. They're really just looking for a value. So rate of oxygen consumption that is different from the rate of oxygen consumption at 25 degrees Celsius. Let's go back to our chart. They already told you that you're looking at 25 degrees Celsius and you're looking at rate of oxygen consumption. So this is really just your ability to point it out on the table, which is this one right here. So rate of oxygen consumption at 25 degrees. They're asking you which rate of oxygen consumption is different. So frankly, there's only two options for this one. It's either this one or this one. You're looking for which one is different, which is a strange way of phrasing it, but they're looking for basically statistically significant, which requires you to know what your error bars mean. So for this one, you have 12.8 plus or minus 2.2. So 12.8 plus or minus 2.2. That means that this can range anywhere from, let's see, so 12.8 minus 2.2 would be 10.6. And then, yeah, that would take us to 15. So that's the range for this one. And then for the 25 degrees, oops, so I'll just put 20 degrees here. And again, you didn't need to write out all of this. I'm just putting the solutions. 25 degrees, you've got 16.5 plus or minus 2.0. And so that gives you a range of 14.5 to 18.5. So these ones actually do overlap because this one goes 10.6 to 15 and this one goes 14.5 to 18.5. So they actually overlap. And what that means is that distance between them is actually not significant. So there's no significant difference between 20 and 25 because their ranges overlap. We need to do it for 30 as well, just to double check. So let's put this over here. So for 30 degrees, you've got 22.1 plus or minus 0.7. So that is going to give you a range of 21.4 to 22.8. And so none of these values overlap. So these values of 30 overlap with your values over here of 25. This one ends at 18.5 and this one starts at 21.4. So these two are significantly different from each other because their ranges don't overlap. So the answer for this one is going to be, you're looking for the temperature at which there's a difference. Your temperature at which there is a difference is 30 degrees Celsius. So that's all you had to put, 30 degrees Celsius. Okay, so the next part says for C part 1, describe on the data in table 1 the effect of temperature on the rate of ATP synthesis in liver cells from toads. Okay, let's go back to this bigger one. So you're looking at temperature and I believe ATP synthesis. So temperature on the rate of ATP synthesis. Okay, so we're increasing temperature this way, and we're not looking at oxygen consumption, we're only looking at ATP synthesis, and it looks like as temperature increases, you're also going to have ATP synthesis increase as well. So 12.6, 16.8, 21.07, that's increasing, and it's increasing as your temperature is increasing. So you can say, for this one, you can say positive correlation, you can say direct comparison. I'm just going to say increase in temp, again, write it in full sentences, equals increase in ATP synthesis. Okay, and then for number 2, it says, based on the data in table 1, calculate the average amount of oxygen consumed in nanomoles for 10 milligrams of mitochondrial protein after 10 minutes at 25 degrees Celsius. So I've seen a couple different answers for this one. I'll put what I personally think the answer is. However, I would obviously wait until the actual marking scheme comes out. But they're asking you for the oxygen consumed at 25 degrees Celsius. And then they're giving you different units. So let's look at it, what it is currently, and then we'll just convert the units is all you have to do. So oxygen at 25 degrees Celsius. So our oxygen at 25 degrees Celsius is this number right here, which is 16.5, and then make sure you take note of the units here because that's what's the most important. Okay, so you've got 16.5. They're looking for average amount, so you don't really need to consider the standard error for this one. So I'm just going to leave that out. I'm pretty sure you can just ignore the standard error for this. Nanomoles per min per milligram. Okay, so nanomoles per min per milligram. Okay, so this is what it currently is. Now they're asking you for 10 milligrams for 10 minutes. Okay, so we're looking at 10 minutes and you're looking at 10 milligrams. Right? You're keeping, and then you're looking for how the 16.5 would change. So for this one, you would multiply it by 10 and then you would multiply it by 10 again because you're looking for here, you're looking at 1 minute, but here you're looking at 10 minutes. So multiply by 10. And then here you're looking at 1 milligram, but now you're looking at 10 milligrams. So then you would multiply that by another 10. I saw some students do it only by 110. However, I believe since 2 of the measurements are changing, you would multiply it by 100 instead of just 10. So your answer for this one would be, so 16.5 times 10 times 10. It should give you 1650. Okay. Animals. Okay. Again, I'm going to wait for the answers to come out, but since you are changing 2 of the units by 10, you would likely have to multiply both of them. Okay. Now, for the last part of this question, part D, the first part says oligomycin is a compound that can block the channel protein function of ATP synthase. Predict the effects of using oligomycin on the proton gradient across the inner mitochondrial membrane. So this one, again, is more of a theory question. I'll add another slide here. So oligomycin is going to block the function of ATP synthase. So it's really important, obviously, that you know about ATP synthase and what the proton gradient does in the mitochondrial membrane, and then you have to justify it. So this is D. Let me draw a little diagram here. So you have your mitochondria like this, and then we have your inner membrane with the cristae. This. Now you have your ETC, which is going to be a bunch of proteins along here. And what they're going to do is they're going to pump H plus from here, from the matrix into the inner membrane space. And that's what builds up that proton gradient. So you're going to have a really high amount of hydrogen in your inner membrane. Okay. Now you build up that gradient for the sole purpose of this protein here, which is going to be our ATP synthase. Your ATP synthase should have the hydrogens come back through, and that's what drives the generation of ATP, right? So you're pumping H plus into the inner mitochondrial membrane, and then the H plus flows back into the matrix through ATP synthase. However, they're saying that oligomycin is blocking ATP synthase from working. So they're asking you what would happen to the proton gradient if this was not functioning. So this is not functioning because of oligomycin. So if you had oligomycin, you would basically have all the H plus build up in that inner membrane and it wouldn't be able to come back through. So there's probably a couple different things that you can say for this. I'll put everything that I think is appropriate. So the H plus gradient would build up in the inner membrane. However, you would not be able to sort of relieve that gradient. So cannot relieve gradient. The H plus can't flow back through the ATP synthase. So that would cause a couple things to happen. So they would have probably accepted any of these. So the pH of the inner mitochondrial membrane would decrease. So pH of inner membrane would decrease. They're asking you more so for the proton gradient. So this is kind of an after effect. I'm taking it a little bit farther, so I'd be curious to see the answer guide. But in terms of the effect on the proton gradient, you would have the proton gradient build up in the inner membrane, and then it would just stay there. You wouldn't be able to relieve it in any way. So you would just have your proton gradient basically constantly on. So pH of inner membrane would decrease, and then you would have less ATP synthesis because that driving of the H plus through ATP synthesis is what powers ATP synthesis. So less ATP. And you basically would not be able to diffuse the gradient or turn the gradient off. Okay, so that's the answer for D for both part 1 and 2. Now, that is the end of number 2 for the AP exam. That brings us to the end of the long questions. Okay, so number 3 of our shorter questions, these are typically, I'd say, 4 to 6 marks. This one says to investigate whether red blood cells of animals lose the ability to take in glucose from their environment as they age. Scientists collected red blood cells from guinea pigs that ranged in age from 1 day old to 7 months old. Scientists incubated an equal number of red blood cells in separate culture dishes that contained a 300 nanomolar solution of radioactively labeled glucose. The amount of radioactively labeled glucose present inside the red blood cells of each group was measured over time. Okay, so A is a pretty straightforward question. It's just asking you to describe a difference between passive and active transport. Now, it's asking you for a singular difference. There's a couple different options that you could say for these ones. So I'll put down the two that I thought of. So passive transport, the most obvious one is that it requires no ATP. You can say it requires no ATP, it's spontaneous, no energy input, anything like that. And since it doesn't require any ATP, it is going with the concentration gradient. So it goes with concentration gradient. Or you could say it moves from high to low, which is with the concentration gradient. And then for active, it requires ATP or energy input. You can say it's non-spontaneous, endergonic, any of those. And then it goes against the concentration gradient. Or you could say it goes from low to high concentration. Any of those would have worked. They were only asking you for one of them. Okay, and then for B, it asks you to justify why the scientist used an equal number of red blood cells in each culture dish as a control. So they're really just asking you to justify why they used that control. So let's go back into the question. So scientists incubated an equal number of red blood cells in separate culture dishes that contained this 300 nanomolar solution of radioactively labeled glucose. So the reason that they would use the same number of red blood cells is because of what they're actually looking at as the experiment. So what they're really trying to look at is whether red blood cells lose the ability to take in glucose based on age. So you're looking at age versus ability to take in glucose. So if you were to plot this, you would look at, again, you don't need to plot it, but you would look at age here, and then you would look at like glucose uptake. So that's your independent, your dependent variable. And so a control that you would have to do is to make sure you're using the same amount of red blood cells in each trial. Because if you didn't, then you would have another variable at play and you wouldn't be able to see directly if age is what's causing that change. So since you want to look at age, so since your like independent or your changing variable is age, so since independent, or you can say the thing that you're looking at, independent variable is age, you have to keep all the other variables constant. So other variables like the number of red blood cells, you also need to keep the amount of glucose that you added as a control as well. So there's a bunch of different controls here. But basically, since you're only looking, like you're only attempting to look at the effect of age, you can't change any other variables. Let's keep other variables as control so that you can like directly look at the effect. Okay, so that's it for A and B. Now for C and D, it says glucose transporters are required for facilitated diffusion of glucose into red blood cells. The scientists claim that the expression of gene encoding these transporters decreases as guinea pigs age. If the scientists' claim is supported by experimental data, predict the effect of increased age on the amount of radioactively labeled glucose present inside the cells of each group, and then justify your prediction. Okay, so you have your red blood cells and it's facilitated diffusion, which means that they just have to go through the transporters. Now, scientists claim that the expression of genes encoding these transporters decreases with age. This is the key. Okay, so if I'm decreasing expression, so decrease gene expression of transporters, you have to mention the fact that that would then decrease the number of transporters. So yes, it's a decrease in gene expression, but we know that a decrease in gene expression is going to result in a decrease in protein level. So you're going to decrease protein level or just decrease like number of transporters. As they age. So as the guinea pigs get older, since they are decreasing in gene expression, they're also decreasing in the number of transporters. So a guinea pig that is, 7 months old would have less transporters than one that is, 7 days old, for example. So they're asking you to predict the effect of increased age on the amount of radioactively labeled glucose present inside the cell. So you've got your red blood cells here, and then you've got your transporters. That would be, here. If it's an older guinea pig, it would have less, and if it was a younger guinea pig, it would have more. And it's saying that the glucose is being uptaken into the cell. So the glucose is going from here into here. Now, if there are less transporters, you would have a less glucose inside the cell. Just because there's less transporters, there's less things taking it in. So if you decrease gene expression of transporters, you decrease the number of transporters as they age. So you can say, therefore, older guinea pigs would have less glucose. In their red blood cells. Okay, so older guinea pigs would have less glucose within their red blood cells because they have less transporters. And the reason they have less transporters is because the gene expression of the transporters decreases with age. Okay, and that's actually it for number 3. So this one was pretty straightforward. Okay, so for number 4, it says the common wild oat is native to regions of Europe and Asia, but is an invasive species in Central California grasslands. In California, the common wild oat has almost completely replaced some species of native grasslands. Researchers found that aphids, a type of small insect that often carries plant viruses, have a much higher reproductive rate in grasslands that include the common wild oat than in grasslands composed of only native bunchgrass species. Additionally, the viruses carried by the aphids appear to affect only the native bunchgrasses and not the common wild oat. Native bunchgrasses infected by the virus have much higher death rates than do native bunchgrasses that are not infected. Okay, so if we start off with A, which is a pretty straightforward question, it says describe the change in the resilience of an ecosystem when there is a decrease in the number of species. So they're really just looking at what happens to the ecosystem when there is a decrease in the number of species. So we've actually answered this in one of the previous questions when we were talking about, I believe, genetic diversity for the crossing over events. So when there is a decrease in number of species, or decrease in genetic variation, decrease in biodiversity, any of those, decrease in number of species, you're going to see a decrease in resilience. Okay, so yeah. You're just going to see a decrease in resilience. It's just asking you to describe, so I don't think you need to mention too much here. If you needed to carry on with this, it would cause a decrease in resilience just because there's less diversity. And when you have less diversity, you have animals with less niches. And so if there were some sort of disturbance like a virus or really anything like a natural disaster, if you have less diversity, it's going to be very hard for that ecosystem to kind of recover, right? Imagine if a virus goes through a ecosystem where there are 2 different, types of species versus, 50 different types of species. They all are going to, like viruses, are going to affect different species at different rates. So the more different types of species you have, the more likely they're going to be resilient. Okay. And then for B, it says, explain how the addition of the common wild oat affects the number of native bunchgrass plants that can be supported by the California grassland ecosystems. This is a pretty broad question. There's probably a couple things you can say for this one. So addition of the wild oat affects the number of native bunchgrass plants. So if we go back to the question, it says that wild oat has almost completely replaced species of native bunchgrass, and then that's because of the viruses. So I would say that the wild oat decreases the number of native bunchgrass plants and the number that it can support. that can be supported. Now, I would probably go a little bit further with this. Now, there's a couple reasons why this would happen. One of the main reasons is mentioned in the question. So that's because the wild oat bring viruses, and that's going to decrease the fitness of the bunch grass. So there's going to be less of them. So either due to the fact that there is, the virus, you could also mention the fact that if there is more wild oat, they're going to take up resources, so take up space. So oat takes space, nutrients, water, any of those. Any of those would have been acceptable, but overall, the wild oat is an invasive species. So as long as you gave some sort of justification for the fact that the number of bunch grass plants would decrease, since wild oat is an invasive species, the question specifically mentions that they bring viruses that are more harmful for bunch grass, but also just the wild oat being there would take up space, nutrients, water, etc. Now for part C, researchers suggest adding ladybugs, predators of aphids, to the California grasslands, predict the effect of adding ladybugs on the abundance of native bunch grass population, and then D, justify your prediction. So I'll just do this one all together. So if the ladybugs eat the aphids, you should see a decrease in aphids, right? So if I see a decrease in aphids, we'll go back to the question. The aphids carry plant viruses and have a much higher reproductive rate in grasslands that include wild oat. So the wild oat plants are basically bringing in these aphids, and the aphids affect the native bunch grasses by giving them higher death rates. So if I decrease the amount of aphids, I'm going to decrease the amount of virus they bring in. And then that means that I'm going to decrease the amount of bunch grass that are going to be affected by the virus. So if I decrease the amount of virus in the population, since it seems that the virus is not only, but majority affecting the bunch grass population, you should see an increase in bunch grass population. Because you're basically eliminating that virus. And so you should see an increase in the population of bunch grass. So if I were to justify this prediction, adding ladybugs, decreases aphids, resulting in less virus affecting the bunch grass. You would basically see an increase in, specifically they're asking for bunch grass, so increase in bunch grass population. You would probably see a decrease in the wild oat population, but that doesn't need to be mentioned. So an increase in bunch grass population, and I think that's all you had to say in terms of justifying it. So they would have basically higher fitness, you could say. Again, I'm just adding on to this. I don't know what the answer guide is. They would have higher fitness, or basically less death rates. Okay, so that is actually just all for this one as well. So I feel like number 4 was pretty straightforward as well. We'll move on to number 5. Okay, so moving on to number 5, this one starts with a pretty basic question. It says, researchers study mechanisms that enable or prevent speciation. A says, describe a post-zygotic mechanism that prevents gene flow and thus enables speciation. Okay, so this is from our final unit, and so post-zygotic would indicate that it is after the formation of the zygote. Now we know that for two things to be considered separate species, they need to be able to reproduce as well as have offspring that are both viable and fertile and all of that. So there are 3 post-zygotic mechanisms that you could mention. I'll put all 3 of them, but you only needed to put one. So the first one is going to be hybrid infertility. And you probably have to go on and give a definition of this. So this would just simply mean that the hybrids that are formed would not be fertile, and so therefore that stops them from being separate species. You would have hybrid inviability. So this would be when the hybrids are at a much lower fitness. So either they die in infancy, they die as an embryo, or they're born but they're not as fit for the environment. So we'll just put here that it would be lower fitness of the offspring. Can't reproduce would be infertility. And again, you only had to put one of these. And then the final one, hybrid breakdown. Now, hybrid breakdown is when they can do the F1 generation, but the F2 generation has the accumulation of too many recessive alleles. So they will begin to break down. And so it would also be lower fitness, but it would take a little bit longer over like multiple generations. Of F2, or you could say over generations. Okay, so that is A, probably just 1 mark, and you could have said any one of these. Now to move on, it says new genes can evolve from non-coding regions of DNA. It is not until certain regulatory elements are present in the DNA that a non-coding region becomes a new functional gene that encodes a protein. These regulatory elements include a promoter, a 5' untranslated region, or UTR, followed by a start codon, and a 3' UTR, followed by a stop codon, seen in figure 1. So they're just giving you the basic structure of a functional gene. This is specific to this AG gene, but all coding genes will have this structure where they'll have the promoter, where you have the binding, the 5' UTR, your exons, your introns, the main bulk of it, which is your AG repeat sequence, and then you always have a 3' UTR. Okay, so let's go to the next part of the question. Zoom this in a little bit. This says, researchers studied the evolution of the family of antifreeze glycoprotein AG encoding genes in Gattidae, a family of marine fish known as cods. When present in the fish, these glycoproteins reduce the freezing temperature of the fish. The researchers compared genomic sequences in 9 cod species and 1 non-cod fish species, B. brosme. They recorded the presence or absence of the elements of functional AG genes as well as AG-like sequences that are similar to a functional gene but have undergone mutation and do not contain all the elements required to enable protein production. And this is seen in figure 2. Okay, so figure 2, you have a phylogenetic tree showing the evolution of AG genes. Now, this might look a little bit overwhelming to students, so I'll take you through this. Here you have your phylogenetic tree. So it's just showing you the descendants. They give you the nodes where all of these sort of arise. And then they are going to indicate to you, which is going to go into our first question, that there was a period of freezing temperatures between 10 and 15 million years ago that you can consider as kind of like a disturbance. So there's a change in the environment at this point here. And then we saw rapid speciation after that. Here is looking at whether or not they contain the functional parts of the gene, including promoter, AG repeat sequences, and 3' UTR. Everything after this point would have 5' UTR and the AG-like repeats because they are after that node. If it has a negative sign like here, it means it doesn't have, in this case, the promoter. A positive sign would indicate that they do have it. They tell you the average water temperature of each fish as well. So you can see that these top ones usually live at about 5 to 10 degrees Celsius, while these bottom ones live at 1.2 to 6 degrees Celsius, so a little bit colder. And then finally, you have this L and this P here. If there's an L beside them, like these ones, it means that they had the gene for the glycoproteins and then they lost them. And it tells you in the question that it was a similar gene but had undergone mutation. And then the P, the ones beside these ones, indicate that it's still a functional gene that's capable of making protein. Okay, so for part B, let's do this one. Part B says, based on the data in figure 2, explain how changes to the genome enable cods to survive and reproduce after a freezing temperatures between 10 and 15 million years ago. Okay, so this one's actually pretty straightforward, and it's not really asking you anything about this diagram here. Just think about it in general when you're changing the genome sequence. So the change of the genome sequence is going to change your phenotype. Okay, and after this period of freezing temperatures, you had a change in the environment, so the temperatures would have dropped. And as a result, the ones that had the mutation that had this antifreeze glycoprotein, because having the antifreeze glycoprotein means that you can survive at colder temperatures. So the ones that did have all of these functional genes were able to survive and would have been, selected for in this colder environment. The colder environment would have selected against these ones over here that don't have the antifreeze proteins. Okay. So it really comes down to natural selection and the fact that having the glycoproteins helps you be better equipped and selected for in this period right here where it did get colder. So for this one, we'll put that the genetic sequence, so it's asking specifically how the genome enables cause to survive. So we'll put genome changes / mutations. We'll say that it reflects the phenotype. So reflects phenotype. So those with the AG genes, which are for the glycoproteins, they would produce antifreeze proteins. And these ones would be selected for by the environment. By the colder environment, so during that period of freezing temperatures. And then you would also probably have to indicate that it would be vice versa. So reproduce after, explain how the changes to the genome enables cause to survive and reproduce after a period of freezing temperatures. Okay, so actually you wouldn't, because they're really just referring to the ones that survived after this. These ones over here did not have the antifreeze proteins, you can see, because these ones are missing all of the elements of the gene, and then these ones mutated to be non-functional. So everything within this 5 to 10 degrees average water temperature do not make those glycoproteins, therefore they're not equipped, or they're not fit to live at 1.2 to 2 degrees Celsius. However, they would have to live at a warmer temperature because that's the only way that they can survive. So we can just put species without, and we can say functional. AG wouldn't survive. So it's just a process of natural selection. Okay, so there's probably a bunch of different ways in which you can say this, but as long as you kept the idea of that the genome is going to change the ability to produce or not produce antifreeze proteins, and that is going to determine what temperature they can live at because of natural selection. So as long as I think you include those key points, you should be fine. Okay, let's zoom in on C here, which says, using the template in the space provided for your response, place an X on the phylogenetic tree to represent the origin of the functional AG gene. Now, I saw a couple students' comments. There was a little bit of disagreement on the answer for this one, so I'll put what I personally think the answer is. Again, we need to wait till the answer guide comes out, but it's asking you to represent the origin of the functional AG gene. Okay. So what's happening here is you're having the emergence of this AG-like gene right here, okay? And then if we're looking, so these ones just completely are missing, all of the functional components of it. So these are not going to include any of the AG functional genes. Which ones do have the AG functional genes, though, are these ones over here. Now, the issue for students here is these two over here. So obviously these ones have functional AG genes. They have this P here, which means that they're functional. However, it's asking for the origin of the initial functional gene. And what they had mentioned in the question happened to these ones that have the L beside them is that AG-like sequences that are similar to a functional gene but have undergone mutation and do not contain all the elements required. So I believe these ones that have the L had functional genes, but they had lost them due to mutation. Okay, so you would still be able to include them in, having the functional gene, but they evolved after the emergence of that gene, and they had a mutation that made it non-functional. So I saw a lot of students wanting to put it right here because these are the only functional ones. However, in my personal opinion, I think you would put the X right here because what had happened is you had the emergence of this gene being functional right here. And then what happened is it remained functional going this way, but it did not remain functional going this way. So the gene was functional at this point, and then you had it branch off to ones that maintain their functionality, and then the ones that mutated to not become functional anymore. I think that's kind of the key to the question, is that the genes were functional and then became non-functional, these ones with the L. So then you would have to put the X prior to that. You wouldn't be able to put the X right here because that would indicate that, this one has a functional AG gene, but it's missing all the components. You couldn't put it here because, again, you're indicating that all these ones have the functional genes, but they don't even have the components for it. So my final answer for this one is that you have to look for the common ancestor of all of these. Definitely the P's because they have, the present functional gene, and then I do believe the L's as well because they had a functional gene and then was then lost. So the origin of it when it originally, came about would be right here. So I would put my X right there. Okay, the final part, D, says, based on figure 2, explain how genetic differences among the species in the Gattidae family determine the habitats in which they can survive. Okay, so this one's a little bit of a strange question because it's pretty much the same as B, so I'd be curious to see the answer guides for this one, but it's the same type of thing. They're asking you changes in the genome versus genetic differences. So for these ones, I believe they specifically want you to address both of the different groups, which is probably the only difference between these ones. So I would go with this pretty much the same thing that I mentioned in part D, is that the genetic differences, so whether or not they have the AG-like gene, is going to determine what temperature they can live at because of they're always going to live where they're best fit. So for this one, I would say that those that do have functional AG gene they would produce antifreeze proteins and then be best fit or best suited, or you could also say selected for, best fit for, or selected for in colder environments. I would make sure to mention the actual numbers. So I would mention in the colder environments 1.2 to 6 degrees Celsius. And then I would say that the ones that don't have the functional gene would not be able to survive in that temperature. Therefore, they have to live at the warmer temperatures because that's what they're most fit to live at. They're in no way fit to live at these colder temperatures because they don't produce those specific proteins. So non-functional AG, they don't produce the proteins. So we're not fit for the colder temperatures. And therefore they must live at warmer temperatures. And then you can include the actual metric here, so 5 to 10 degrees Celsius. Okay, so I think for both B and D, as long as you mention, the fact that it reflects the phenotype and talk about how this is reflecting the ability to actually produce these special proteins, and the environment is going to select for or against the fish species based on whether or not they can produce these proteins. Okay, if they can, the colder environment will select for them, and if they can't, the warmer environment will select for them. Okay, so that's the end of 5. Okay, 6 says that scientists can quantify the rate of translation as ribosomes move along an mRNA from one codon to the next using a procedure called ribosome profiling. The scientists measured how long a ribosome remains stationary at each codon of each mRNA. They determined the average translation rate across all codons is 5.2 amino acids per second, but that the average translation rate of specific codons in different mRNAs can vary widely. These variations in translation rates are thought to facilitate correct folding of the protein being produced. The rate at which 3 different codons were translated was measured in 100 different mRNAs. The scientists determined the distribution of rate, so the number of times each rate was recorded. For each of the 3 codons, you have GAC, AUU, and UGG. Okay, so these graphs are a little bit different from what you're used to. They're not the standard bar graphs that we made in, I believe, question 2. These are more so what are known as histograms. And so what histograms are they're really just the count of how many times a certain instance occurred. So the amount of times that they recorded, 150 milliseconds per codon was 20. So they got 20 times, so they got that rate 20 times. So this is more so a histogram for count. And the important thing about this one is to make sure you're looking at the units, because I had some students read these graphs kind of the opposite way that you should. But if you just paid attention to the units that they gave you, it would indicate, what you're supposed to be looking at. So you've got these numbers down here on your x-axis, and it's in milliseconds per codon. So this would be 150 milliseconds per codon, and then over here would be 650 milliseconds per codon. So over towards this end is actually the faster, and then towards this end is slower. Okay, because this indicates that it takes less time to translate one codon. I had some students, if you read this incorrectly and thought it was like codons like per second, which I believe they kind of reference they reference that in the question. However, you have to be mindful of what they actually give you in the graph, which is milliseconds per codon. However, if this was 650 codons per second, then it would be opposite, where the opposite side is considered faster. But since it's milliseconds per codon, everything closer to the left end of the graph is going to be faster, and then everything to the right end is going to be slower. Okay, so if you were confused by this one, just keep in mind what the units actually are and what the bottom x-axis is indicating. Okay, so let's go into our first question. It says, using data in figure 1, graph A, identify the rate in milliseconds per codon that was recorded the greatest number of times for the GAC codon. So figure 1, graph A, we're looking for the most recorded times. So this is this graph over here. If you zoom this in, you're looking for the rate that they want you to report it in milliseconds per codon. So you're looking at the x-axis. Which one of these was recorded the most amount of times? Okay, recorded the greatest number of times for GAC, and our top graph is GAC here. So we're really just looking for the highest number that was recorded. The highest one in here is this bar right here. If you look at what the rate is, it's 150 milliseconds per codon. So the answer for that one is very straightforward. You would just put 150 milliseconds per codon and make sure to put your units. Okay. And then part B says, using the data in figure 1, graphs B and C, describe the variation in translation rate of the AUU codon compared to the UGG codon. So we're looking and comparing B and C. Okay, so this is where it's important that we kept in mind which side of the graph is, the faster rate and which one is the slower rate. So if we look at B, you have majority of the recorded events happening, between 50 and 200 milliseconds per codon, which is relatively fast. And then if you look at UGG, it's much more spread out, and you have it ranging anywhere from like 50 to 950 milliseconds per codon. But on average, this one, it's more towards the other end if you were to average it out. Now, for this question here, they're asking you to describe the variation. Now, for this one, Variation usually is going to indicate, in my opinion, the spread of the graphs, because they do have very different types of spread. For this one, I would probably mention spread as well as on average which one is faster, just in case. So I would say that AUU, so our graph B, is less spread out, or you can say less variation. More specific is another way you can put it. And then you can say compared to the other one, compared to UGG, on average, and I would mention on average, because you do have obviously a spread and you have multiple numbers, on average, faster translation rate. Now for UGG, it would just be the opposite. Okay, so more variation, because we saw a really big spread of that data, it probably would have a very large, like if we're thinking about it in the fact of like significant error, and then on average, slower. Okay, so that would be B. You're really just reporting the values that you're getting. C is scientists hypothesize that tRNA molecules that bind to UGG codons are available in lower abundance than tRNAs that bind to AUU codons. Support the scientists' hypothesis using the data in figure 1. Okay, so this is why I think B is more so referring to spread, because C is referring more so to rate. So, which one's faster and which one's slower. They're saying that the UGG codons are available in lower abundance. That tRNAs that code for them are in lower abundance. We know that the tRNAs have to bring the amino acids over to the codon to obviously do translation. So if there are less tRNAs available, it's going to slow down translation, right? It's like doing a project with 5 people versus doing a project with 500 people. If you basically have less workers, it's going to take a lot longer. So that does make sense with our data, because when we looked at UGG, we said on average the rate is much slower, because you're getting most of the bars around this region here. I would say maybe on average about 350 is where that middle part is, whereas here you're getting the highest amount around 100. So it's very clear that this one is going a lot slower, like we indicated in part B. I would maybe re-mention it. So it does make sense with figure 1, because UGG has slower average translation rate. And so since the translation rate is slower, it's likely that there are less tRNAs, likely less tRNAs specific to UGG, limiting rate. Okay. So you really just had to justify the fact that in figure 1, you can see that UGG is slower, and UGG being slower does make sense if it does have a lower abundance of tRNAs, right? It's going to take longer to find the right tRNA and bring it over, which is going to slow down translation rate. Now the last one, D, says amino acids can be encoded by multiple codons. In many organisms, certain codons for the same amino acid occur more frequently than in mRNA than do other codons. Based on the data provided, explain why the use of one codon over another for the same amino acid might result in increased level of protein production for a particular mRNA. Okay, so this really just goes along with C. I'll put it right here. We'll put D here. Explain why the use of one codon over another for the same amino acid would result in increased levels of protein production. Okay, so I would say that for this one, we can clearly see that just based on these 3 graphs, that codons have different rates of translation. Okay, so codons tend to have different rates of translation. And to justify that, or to give an explanation why, I would say because, they're limited by how many tRNAs they have. So codons have different rate of translation because of tRNA availability. Slower translation. So they don't actually, you have to make the connection between rate of translation and protein levels. So you have to say that slower translation would result in decreased protein levels, okay? So since codons are different in how fast they translate, they're also going to be different in how much protein they're able to produce in a given amount of time. So for example, UGG, our slow one over here, is not going to be able to produce as much protein in a given amount of time when compared to AUU, for example. So the protein expression is going to be a lot lower in UGG just because it takes longer to make the protein. So slower translation means decreased protein levels. So you have to make that connection. I feel like that's where you'll get the point. Decreased protein production. Okay, so as long as you connected those two, I think you should get the mark for that one. Okay, so that's actually the end of our FRQs. We did all 6 of them. If you have any questions, please let me know in terms of, if you had different answers for these and you want me to check them, or if you're wondering what counts and what wouldn't. We will have to wait for the actual marking scheme to come out so that I can give you guys 100% answers for these, but this, in my opinion, are the correct answers for your FRQ questions for this past AP exam. Okay, thanks guys.